Hello, everyone. Thank you for joining in, and I hope everyone is in good health and are doing good. So, today we have gathered for the session of Psyomics. It's a student club for bioinformatics. Subhajit Barua, my colleague, is the president of Psyomics Club, and I myself, Rutvi Wajai, is serving as the vice president of the Psyomics Student Club. Our community comprises of more than 1,000 plus students, 20 plus researchers, and many more mentors and peer reviewers who are a part of our Psyomic Student Club community. Together, we meet for sessions, discuss and brainstorm ideas, and take the concepts of bioinformatics at a next level in order to discuss its application in various other fields. What do we do, and the topics that we focus on are some of them are infectious diseases, microbiomes and microbe, precision oncology, plant science, and neuroscience. One of the main goals of Psyomic Student Club is to help students and other people clear the concepts of bioinformatics. Not only this, but we also help the students to gain insight on how to use different tools of bioinformatics, and not only different tools, but free different tools. We also have pipelines, and we have like a hands-on workshop and help them to learn how to apply the research using bioinformatics in day-to-day life. We also help some students who are interested in the coding part of bioinformatics to learn the application of coding in bioinformatics. However, these data science skills together help the students to apply the bioinformatics knowledge in different areas stated over here, such as neuroscience, cancer, microbiology, and infectious disease, and many more. Talking about what OmicsLogic is. So OmicsLogic platform is a platform whose main goal is to provide training in bioinformatics in order to enable independent research, which is guided by mentors and peer example who have more than a great years of experience in academia, industry, and research. We are developing a growing community with shared passion for data-driven research and appreciation of citizen science. We help students and clinicians, along with faculty of all backgrounds, to develop novel and independent research projects using technology in life and data sciences. Speaking about one of our greatest upcoming events is the OmicsLogic Research Symposium 2022. It is from March 31st and April 1st, 2022. The main goal of the symposium is to bring the peers, the students, the mentors, and the faculties, researchers, and academicians together to form a platform and interact about ideas and ongoing research related to bioinformatics, education, computational sciences, and further on. I myself was a part of this Omics Research Symposium last year, where I had a poster presentation and I won the Citizen Project Prize for the same. So this year also you guys can participate in the poster presentation competition, whose details would be shared in the chat box. You can click on the link and submit your abstract, and the further details would be shared in the upcoming slides. As an experience, and talking about the experience I had for the poster presentation was immense. I got exposure to so many new people I haven't met. My project got reviews from one of the best universities in the world. There were speakers from Georgetown University, Blue Marble Space Academy, and they gave me reviews on my project, which helped me improve further and showed me directions about how to improve and work on your project and in which direction to work with. Overall, it was very helpful learning and improving experience. Not only about this, but you also make connections which help you to form deeper connections or find your pathways, or in what field you should go to, and advisory kinds of connections. So the symposium topics for 2020 Research Symposium is new advances in enabling technologies, career opportunities, and the poster competition that I talked about. We have academic experts from Georgetown University, Tauber Bioinformatics Research Center, Google, Faber Academy, Ames Research Center, Blue Marble Space, and we have experts from our network partners such as LBRN, HA3 Bionet, and further on. Talking about the topics that would be covered in Omics Research Symposium, and our confirmed speakers includes Google. So they would be talking about Google Cloud Resources and the NIH Strides initiative. We have speakers from MIT University, which would be talking about bioinformatics education and research training at MIT University, Noida. We have speakers from Blue Marble Space, which is one of the renowned institute for astrobiology, and they would be talking about careers and internship opportunities in astrobiology. We have another speaker from Google, which would be talking about Google's partner programs and resources. Similarly, there are furthermore confirmed speakers, and if you want to get involved in this kind of omics research symposium, then as a student, I'm saying from my side that one should not miss this, because it's one in a lifetime experience, and we do not get listening to such speakers on a daily basis. So for the poster presentation competition, there were these three posters that won the first prize, second prize, and the best citizen science project prize in the last year. You get amazing prizes and the exposure to the peer reviewers and the industry experts, which is something we do not usually get in all the conferences. The conference's poster presentation themes for this year are omics and bioinformatics, artificial intelligence, machine learning and data science, astrobiology and space omics, and computational biology in biotech and pharma. We have prizes for Best Poster Presentation, Best Citizen Science Project, and Young Sheldon Award this year. So starting with today's session would be conducted by Dr. Mohit Mazumder, who is the Business Development Director and Project Mentor at Pine Biotech. Today, he would be speaking about structural biology and research project designing. So hello, Mohit sir, the stage is all yours. Thank you.
Hello, everyone. Hi, Ruthvi. Thank you for this wonderful introduction. And I hope that many of you would talk about this session or talk about this initiative called SciOmics and help others to join as well in the future. So today we planned to discuss that we will discuss about structural biology and research project designing. So one of the reasons for that is obviously the upcoming symposium, because it is an opportunity where you can showcase your poster presentation or presentation. So in that sense, it helps to incorporate good images and good analysis using maybe if you are interested in structural analysis, and I guess many of you are, that's why you are joining this session or joining for this session to learn. So I hope that many of you would do that. So a very good morning and very good evening to all of you, wherever you all are. So today we will be covering few important aspects of structural biology analysis or, project designing. So let me share my screen. Just a second, please. So yeah, so as I was saying that I wanted to ask all of you before we start actually that how many of you have like prior experience in structural biology or using computational structural biology, or like how does this interest comes in? So what is that you want to do in structural biology? So you can write some of this in the chat. I'll be happy to know and learn about your interest, and then we can discuss some of those things as well. So please feel free to write in the chat, and we will have maybe discussion towards the end of this presentation. So all right. Yes. So today, in today's session, what we are going to do is that we are going to have an overview of the very basics of structural biology. So that's why I also wanted to know that who have this experience in structural biology, so that I would know that how much depth I have to go. And if you're a beginner in structural biology, please let me know in the chat that I'm a beginner and I would want to go from the very basic. So I mean, from structural biology, I want to discuss about also the current application of structural biology and how it could be, applied in the context of a research project. So that would involve, understanding how this field has evolved, what different things that we have learned so far, and what is in application. So what is that is used right now in the industry, in the research labs, and then also like from the public data, how do we think about doing research projects or research that is possible, so through these different tools that we are going to review or like talk about. So in today's session, we are going to start with an overview, very basic overview. Then we are going to discuss a research project case study. So that case study would have several aspects of research project designing. So that's how we are going to cover some aspect of that through the case study. And then we will also go into some understanding of how, like, from sequence to structure to function, we will try to understand all of that in the context of the case study, and also like what is the downstream analysis after that. So and then we are going to look at some of the new advancements. So we are going to look at some of the things that have happened in the recent few months, in the last few months, and what insights that we can gain from such studies, and how do we design our own research based on some of the information or some of the practice that people in these publications have done. So we are going to review a few publications, and then we are going to discuss about a little bit about, how do we do the setup, how to set up a computational structural biology experiment, such as, docking. And then we are going to discuss about some of the other methods. And after that, we are going to talk about the upcoming events, one of which is the symposium, which we are very excited about. And we are hoping that many of you would participate in there, and also submit your abstract and posters to compete. So yeah, that's how that's the plan. And now let me look at the chats. Just have to beginner. So there are a lot of beginners here. That's very good to know. And also please feel free to write like what degree you are in, so that I'll understand that what kind of things that we can do together. So this is also an opportunity. So towards the end, we are also going to discuss opportunities, and especially for those who are like a little bit proficient with this, or would want to become proficient with this. And then we will also have like opportunities such as to work on project, and also like internships and all. So I hope that this session would be helpful for all of you, and I look forward to have a more like of interaction throughout this session. So please feel free to keep on writing in here in the chat, so that we can also discuss the things that you are not getting to understand, or the questions that you have. So now, if you are a beginner, then let's start from the basics and let's answer a few questions. So structural biology. How do you define structural biology? So structural biology, sorry, I'll have to just stop my video because of the resolution. So because there's a lot of images. So yeah. So structural biology is the study of how biological molecules are built. So we are looking at structures. So it's in 3 dimension. So I think many of you are also many of you are also fascinated to understand that how a three-dimensional structures can be analyzed or could be understood, and also in the context of how fascinating it is from two-dimensional or from sequence to structure, and from the structure to so many different functions, and multimerization and complex formation is really a fascinating way that these molecules work together. So also the key basis of our existence. So as I was saying that using variety of imaging techniques, scientists view molecules in 3 dimension to see how they are assembled, how they function, how they interact, all of those things. So that has helped actually researchers understand how the thousands of different molecules in each of our cells work together to keep us healthy. So this is one aspect, like that structural studies. Because of the structural studies, that has shown that how mishappens molecules also like makes us sick, and as a result of these studies, uh, mm, we have prompted new treatments in many diseases. So I'm talking about not only understanding the way how things work, but also understanding when things do not work properly. So understanding the mechanism here. So today we are going to dive into some of the mechanistic studies, and I think that is important in structural biology, not just understanding the tool, but also the science behind it, the research behind it. So this is kind of a research oriented session. And I hope that many of you would appreciate or try to understand that how to think about it and how to conceptualize to come up with ideas or do contribute in some of these things through analysis and reporting and imaging and all of that. So the first question that you can see on the screen is, what are molecules? So what are molecules? Well, molecules are a group of two or more atoms held together by a chemical bond, right? So molecules include DNA, RNA, proteins, and then carbohydrates, sugars, lipids, which is fats. So structural biologists especially are particularly interested in proteins because they do so much of work. They do so much of work in our body. So that is one of the reasons that many structural biologists, and there are so many structures of proteins that you will find. So increasingly, biologists are investigating large molecules that are made up of a combination of RNA and protein, that are called RNA protein complexes. So what are proteins? The next question. And if you know the answer to some of these questions, please feel free to write it in the chat, and that would be great. And so proteins, what are proteins? So proteins are molecules that contribute to virtually every activity in our body. So they are involved in everything. So they form hairs and fingernails, carry oxygen in the blood, allow muscles to move, and this will be an endless list. So next question, what are proteins made of? So proteins are made of long strands of small molecules called amino acids. So amino acids, what are amino acids? Yes, amino acids are those small molecules with a certain characteristics because of their chemical formulation. So there are 20 amino acids found in nature. In each protein contains a unique combination of few dozens to many thousands of these amino acids. And today we are going to review some of these proteins formed of some of the stretch of amino acids and fold in a specific way. So some protein consists of multiple amino acid strands wound together. So that's about that. And now, the next question, a very important question, is that how does a protein get its shape? So we are discussing today structural biology of macromolecules, and proteins, and even though proteins are a string of amino acids, they do not remain as a straight line. As you know, the strands twists, bends, and folds into specific shapes. The way they fold depends in the part of the way the amino acid interacts with each other. So in our case study today, we are going to see that how that interaction impacts the structure, and how there are, if you replace some of those amino acids, that is like protein designing, how that impacts the structure then, and how can you predict that. So that's our case study today. As I was telling you that some of these proteins are they form standard motifs. So motifs are like corkscrew like coils or called alpha helices, and flat sections are called beta sheets. So these are like motifs are defined based on their conservation. So researchers can easily determine the protein's amino acid sequence nowadays. So the trick has been to figure out how and why protein fold. So scientists are beginning to solve that puzzle with research into how amino acid interacts, and they are using new computer programs that can help predict protein motifs. And many of you are aware of AlphaFold, and we did cover it in the last session also, that how AlphaFold works, and how it has changed the revolutionized the protein folding problem. However, I mean, there is still a lot that is required. So we don't know the structures of all the proteins, and many of are noble proteins, and we don't know how they fold. So obviously all of this information has been possible through experiments, and those wet lab experiments include some of those techniques that I'm going to talk about in the next slide. So the next question then, why does a protein shape matter, right? So a protein structure allows it to perform a specific job. Look, it's to do a specific job. That's why it has a specific shape. So for instance, antibodies are shaped like a Y. That helps these this helps these immune system proteins to bind to a foreign molecule such as bacteria or viruses with one end, while recruiting other immune system proteins with the other. So the design is made in a way that it does its work. So DNA polymerase that you can see on the screen, DNA polymerase 3, is a donut shaped. This helps it to form a ring around the DNA as it copies its genetic information. And the proteins called enzymes have grooves and pockets that help them to hold onto other molecule to speed chemical reactions. So similarly, misfolding or mishappening causes diseases in proteins, They often stop working properly and can build up in tissues. So Alzheimer's diseases, Parkinson diseases, cystic fibrosis are some of the examples caused by misfolded proteins. So I hope that this basic introduction is helpful for many of you, and I know many of you are already aware of this. So the next thing that I wanted to discuss is what kind of proteins are out there, and how do scientists use protein structure to develop new drugs? What are the examples of medicine developed using structure-based drug design? So because we are talking about structure, one of the main utility of understanding the structure is also to develop or design strategies to overcome some of these challenges related to diseases and all of that. So structure helps in that because it gives you precise information of that molecule. And then obviously you can manipulate and design based on structure. So right. So yes, and then like very basics of how do you determine structures? What are the different ways you can determine structure? One of the latest technology, what is a CryoEM? How does that work? So we are going to discuss all of that in the slide. So let's start with the fact that that there are many proteins and there are many different kind of proteins. For example, many proteins are enzymes that bind that aid in biochemical reaction. Others have shapes or functions that can help cells hold their shape and move as described in the previous example. So another type of protein are like transporter proteins. They are embedded in the cell's outer membrane and form channels that help vital substances such as sodium or potassium to pass, sodium, potassium, calcium to pass in and out of cell. So there are different proteins which have different functions and different, there are involved in different pathways. So how do scientists use protein structure to new develop? So obviously drug typically work by either blocking or supporting the activity of a specific protein in the body, right? So using an approach called structure-based drug designing, scientists can make a template of a protein and use that blueprint for creating new medicines. So they start with a computerized model of protein structure, and then they study it further. So this computerized model of protein structure actually comes from an experimental study. So how that happens and what are the algorithms involved from, from a crystal structure to a three-dimensional structure is something that many of you might know. But for example, the computer model allows researchers to examine how two protein could work. And if scientists want to turn off one protein, they can design and alter a molecule that can block or alter that interaction. So these are some of the ways scientists do design new drugs. So, for example, like what would be an example of medicine developed using structure-based drug design? So, yeah, so I think researchers used structure-based drug design to develop some anti-HIV drugs. So HIV protease, I think, is an enzyme that keeps the virus alive. Knowing its structure allowed researchers to determine the kind of molecules that could stop HIV proteases from working. So scientists use computer models to fine-tune these molecules that could halt virus production. So this work led to a medicine called protease inhibitors, which is available. So how do scientists determine protein structures? Now, again, a very basic question. So researchers used several imaging techniques to determine the structure of proteins and other complex molecules. So cryo-electron microscopy, CryoEM, allows scientists to see individual proteins as well as large structures such as molecular complexes, groups of protein that combines and function as a unit, viruses or organelles. So bigger structures can be visualized with cryo-EM, X-ray crystallography, and NMR, so nuclear magnetic resonance spectroscopy, also make it possible for researchers to view proteins, and we will see one of such case study today. So to date, researchers have used these techniques to unravel the structure of more than 1,22,000 some structures, some proteins, and they are available on the protein data bank, where you can access them, download them, and visualize them and understand them better. So, okay, now the next question that I put together for you, as you're not asking yourself, so I'll also look at the chat just for a second. All right. So we were talking about cryo-EM and X-ray crystallography and NMR. So in cryo-EM, researchers rapidly freeze a cell, virus, molecular complex, or other structures so that the water molecules do not have time to form crystals. So this preserves the sample in a natural state, and then scientists use an electron microscope to blast the frozen sample with an electron beam. And this creates a two-dimensional projection of the sample on a digital detector. And by creating hundreds of projections of the sample from different angles and taking the average of those angles, scientists generate a three-dimensional model of the structure. So recent advancements in cryo-EM did provide a highly detailed image of proteins and other biological structures, including large structures such as RNA protein complexes. Another technique is X-ray crystallography. So X-ray crystallography, how does that work? So X-ray crystallography shoots a beam of X-rays through a tiny solid crystal made up of trillions of identical protein molecules. So one crystal has like a lot many of protein molecules, and that crystal is placed in a way where there is a beam hits it, and the crystal scatters the X-ray onto an electronic detector, similar to the way images are captured in a digital camera. So a computer then gauges the intensity of scattered X-ray to assign a position to each atom in the crystallized molecule. So this results in a three-dimensional digital image. So this method has been used to determine like 85% of all known protein structures, and very, very important technique. So another technique I wanted to tell you all about is NMR spectroscopy. So NMR spectroscopy works using natural magnets, the nuclei of certain atoms, including proteins. So these natural cellular magnets interact with a big magnet inside the NMR machine. The big magnet forces the protein magnets to line up. Researchers then blast the sample into a series of split seconds radio wave pulses and observe how the protein's magnet response. So scientists use several sets of these NMR blasts and combine the data to get a more complex picture of the protein. So although X-ray crystallography can examine larger proteins than NMR is able to do, NMR has those limitations that the sequence is usually 100 to 250 amino acid long. I mean, less than 100 is also fine, but that is the range that usually NMR works really well. So NMR technology can be used to study protein immersed in liquid solution, in contrast to X-ray crystallography, where you need to crystallize the proteins, and that is one of the challenges that is there to for crystallographer to crystallize protein, because it requires an intensive process of screening and trying different screens, and then it's very difficult to get crystals, and usually it takes you to do PhDs and work on long projects to get from cloning and purification to crystallization. And many of you who have gone through that process could not agree more, right? So right now, I mean, how scientists view how proteins act. That's an important question. So, I mean, new technology is beginning to allow researchers to progress from creating static pictures of, from static pictures of proteins to now making movies, three-dimensional structure and three-dimensional movies with their actions. So images provide snapshots of what these cellular events are doing at a specific point of time. So if I'm talking about crystal structure, and even if I'm talking about any of these structures, so it's like a picture, right? It's like a photograph that you have clicked. It's not a video. So in a video, you see how a person is moving. You see all about other features that you do not understand in a single snapshot. So that's where, I mean, other techniques comes in. So with a known structure, so if I know how a person, how tall is a person, and if I can model that person, and if I have a three-dimensional model of that person, and if I know the laws of physics, how a person moves, so maybe I can try to model the movement, that how it could be moving in a specific kind of an environment. So I can mimic that environment, and that's how we do molecular dynamic simulations. So as I was telling you that, right, so applying these and getting this information and understanding this dynamic system is critical to unlock how life works, because it is always dynamic. We are always moving. it's always something happening. So in addition, there is a whole class of proteins which are called intrinsically disordered proteins. They do not hold a specific shape. So many proteins, they do not hold a specific shape. They stay disordered. Their shape adapts to what's going on inside the cell. So there are signals making it nearly impossible. I mean, there are signals when they're a part of uh, um, domain in that protein get structured. So, and that is based on certain signal, and it is really difficult to capture those and get them crystallized, or get them to view them or to picture them. So scientists are now using powerful computer models to make molecular movies so that they can see the full range of proteins in live action, by feeding this information from various imaging systems about how amino acid interacts, how building blocks interacts. Now it's possible. So that is one of the things that I have been also doing, is to be able to do simulations and to learn about some of these interactions, to validate some of these interactions, which is kind of the last step. So again, like, what is the future of structure biology? So we will end this kind of overview section by talking about the future. So researchers on the frontiers of structure biology are merging all kind of imaging techniques. So it's a convergence of pictures, X-ray crystallography, NMR, cryo-EM. This allows all of us to create a more precise map of what proteins and other molecules look like, how they interact, because the interactome, the way different molecules are interacting, we are getting more evidences from these scientific literatures, studies, and from connecting those and connecting them through structure, scientists can create a single image that zooms in to see the specific proteins and zooms out to see how they interact within the largest cellular structure, right? So, in addition to combining existing techniques, scientists are also developing more powerful techniques. For example, new X-ray lasers allow insights into the processes that occurs in less than 1/10 of a trillion of a second, much faster time scale that captures by other sources of X-rays. So through X-ray lasers, those are very spontaneous processes, can be also captured. Scientists use super efficient methods now to determine protein structures more efficiently. I told you about AlphaFold. Then the whole application of deep learning. So deep learning has contributed to major advancements in predicting protein structure from sequences, a fundamental problem in structural bioinformatics. So with predictions now approaching the accuracy of crystallographic resolution in many cases, and also let me also mention this, that this has been also possible due to GPUs and TPUs. So these GPUs are, NVIDIA graphical processing units that transform this ability for computers to do so much high-end computing, and it's kind of affordable for labs also. So that helped in fast genome level structure prediction. So not only just predicting one structure, but predicting the entire proteome of individual. So that sort of scaling is required, and that has been come through the technological advancement and is it's coming up. So this is possible through state of art deep learning models, and obviously wealth of new data for system biology applications. So yeah, that's
Excuse
how
me,
we conclude
Dr.
this. Right.
Dr. Mohit, I think your slides are not changing due to some reason.
Okay, I will stop sharing and then I will share again. So yeah, I was concluding this part of the session where we discuss about the overview, and it is meant for all beginners to learn about some of these aspects going into the next part of the session. So next part of the session is kind of special because it's my PhD thesis work, and that's the case study also. So okay, so we don't have a lot of time, so let me quickly do that. Yeah, there is some issue with sharing. Okay, so next part of the presentation, which we will be discussing, is a case study. So from sequence to structure to function to prediction to design. So some of the objectives that we are going to cover is how designing a workflow to predict calcium binding sites in EF-hand proteins, and understanding what protein family is, how hammer is used, 3D structures. Then the second part of this objective, second part of the about this case study is predicting the calcium binding affinity to facilitate protein engineering, which is to do data mining, hypothesis testing, and machine learning. So this is all in the respect of structural biology, and how this is playing, how from sequence to structure to function. This is a case study. So briefly to tell you that what the objectives are is to predict this site that you see on the screen. So on the left side, you see that there is a hand that shows with two thumbs, which denotes two helices, and in between there is a loop where you can see a Ca, which stands for calcium. So calcium is found to be is seen to be binding in this EFN motif, and this especially into this groove, where you will see that it forms a specific kind of a geometry. So that geometry is maintained through binding to the residues at 1, 3, 5, 7, and 12th position. So these are important positions in the respect of this study that we are going to discuss. So in this case of full residue canonical EFN, and this could be seen in molecules such as calmodulin, and I don't know, many of you have heard of this molecule. It's a pretty important molecule, which binds calcium primarily via side chain carboxylates. So the loop sequences, as I was telling you, 1, 3, and 5, 12 are the positions from where it gets to those oxygen molecules from the carboxylate, and then it composed of a highly conserved like this loop. So this loop is something that we have to predict from the protein family. So this is the below you will see that there is a list, there is a graph that has residues written into it. So it signifies the frequency of these amino acids found in the entire family. So an entire family is like sequences from different organisms across, and then that is where you can find those families in PFAM, is one of the famous popular website, where you can download those families, and you can use hammer to see if you to see and find that if your sequence has those proteins or those protein families present or not. So if you're scanning genome wide or like that, many users use that technique to scan for protein families. So in this example, I mean, you will see that there is a specific pattern that I'm talking here about. So this big D that you see is at the first position, and it goes till the word E before the FNL. So it's a 12-mer stretch. These 12 residues are responsible for binding to calcium, and they play an important role in holding the calcium ion there. And based on their strength, you can determine if the calcium is going to bind with a high affinity or a low affinity. So there is some correlation between the sequence and the binding affinity of calcium with the sequence. That is something that we want to predict. So here the third objective is to predict calcium binding affinity. And the hypothesis here is that the sequence has to play a role with the binding affinity. And if we change and vary the sequence, the binding affinity is also going to vary. So, I mean, in what context are we saying that if the sequence is more conserved, if these preferred residues at these positions are available, then it will have a higher binding affinity. So that's the hypothesis that we started with. Now, the next step is to develop flowchart or develop algorithm to find this, to do this like one after another. So pipeline. So we developed two pipelines. One pipeline is to predict that 12-mer. So this is 12-mer that you can see from D to E, and you can see that there is a good pattern here. So if my machine learning algorithm, where I'm going to train these sequences that are already known to bind calcium, if I can model this and tell this machine learning model that, look, these are the places, these are the sequences that are highly conserved, and please look for these sequences and let me know if you find some. So that's the first model. And to do that, I mean, this is the workflow. So first, from the big sequences, so the input, the user, I mean, if someone wants to find EF and 12-mer binding site, how to facilitate that? A person who is going to look for a specific stretch is going to submit from a big sequence. So a sequence could be a protein sequence, a sequence could be a whole genome or whole proteome. So yeah, that is your input. So input is big sequences. So that big sequences is now broken down into 12 mers in a scanning way, so that so that the algorithm can can work and predict from the 12 mers that is being generated through the first step. So but the first step is to break it into 12-mer sequences, which is the size of the active site. Now the next step is to convert that alphabetic order that is there present into machine learning input, which are numbers. So we developed which we used the pattern based recognition, which is like binary. So in binary, what you do is that you assign these 20 amino acids with their own unique patterns. So for example, if it is 1000, like 20 numbers are there, and you keep on changing the position of one for 20 positions, then you have a pattern for binary. Similarly, amino acid composition is one of the features. So these are some of the features that are taken to develop it into a machine learning model. Now, after the machine learning model is developed and tested and validated on different data sets, you figure out that how it is working, how is it being able to predict the sequences or the binding sites correctly or not. How do you validate that? You can validate that by downloading all the structures that are there in PDB that binds to calcium, and look at those binding sites, and then you can test your algorithm by checking one of the sequences if it is correctly predicting the binding site or not. So that is the first hypothesis. Second hypothesis is that what is the correlation between the sequence and the function, which is the binding affinity. So how do you predict the binding affinity of a sequence and from the sequence and relate it to some experimental value? So you need obviously some correlation. So that is what I'm going to tell you about the next slide, in our next slide. So with the literature, so next part is to do literature review. So when you're conducting, or when you're kind of starting an experiment or research, first you need to find where the data is, and then you do some exploratory analysis and you see the pattern, and based on that pattern then you go to the next level and start designing your machine learning. So here we did a lot of combination permutation, and that involves amino acid features. So what the input sequence is, remember the input sequence is amino acids. So amino acids have to be converted into different type of, based on their different features. So amino acids have also different, maybe charge. All amino acids have different charge, so you can differentiate based on their charge. And similarly you can differentiate about with their hydrophobicity and all of those features. So these are the features that have been inputted into the machine learning, that based on these features you classify if they are following a pattern or not. Two machine learning models were designed and created. One is to predict the binding site. So your input is a protein sequence and the binding sites are predicted. Here you can see 4 binding sites are predicted with a SVM score. And the second part is to predict the binding affinity. So if it is predicted the binding site, next what you want to do is to know how strongly or how loosely it's going to bind to calcium. All of this through automation. You just have to get a sequence, and you're getting all this information through automation and you through all this analysis, and that's the project. So the first paper showcased all of this. We published this and published with 100% accuracy for prediction of the binding site, because you see there is a high level of this pattern is very good. So the model was able to easily distinguish between binders and non-binders, basically based on these features, that aspartate is being seen at the first position a lot of times. And if you mutate this aspartate to alanine, you will see that the binding affinity is either lost completely or it's very loose. So this is an important residue. So these are the features that the machine learning model through. And then what we did, like we have created this model and got published and all of that. The next thing that we did was to design a calcium binding site which will have a high binding affinity based on this hypothesis. So we looked into all the binding sites again and looked into their affinity, and all of that literature review was done. And then we found out, then we thought of designing a protein which will bind calcium very strongly based on our hypothesis, based on our algorithm. So the algorithm also gives an output score with each prediction. So if the score is high, the binding is high. And if the score is low, the binding is low. So based on that, and based on the things that are available in the lab, which is the anti-amoeba histolytica EHCABP1 protein, we were able to do this experiment where we mutated one of the protein and made it to bind with high affinity with calcium. So our hypothesis was that if we change these positions that you see here in the D panel of the left side of the image, is that this is the designed one, DK, DG, DG, FI, D, FEE. So D is aspartate, and like similarly, these aspartate is kept as it is at the first position, and then alanine is changed into lysine, and then similarly there are other mutations where phenylalanine is added, and also like these were like not very drastic mutations, but those mutations were something that were not done before. Anyone did not thought of ever to think about like how these residues are going to play a key role in this design molecule. So what we did was a lot of structural analysis, and that's why I wanted to also show you like what are the different ways you can do these visualizations in your project. So these are the different ways you can visualize. So we found out that the protein structure changed, and there is a movement, 7.1 degree movement in the mutated protein when we compared it with the native protein, and we like mutated it and then crystallized it, and then found out that there is a huge change in, there are slight changes in the active site that made this entire protein do a lot of different things. So from a trimeric protein, it became a hexameric protein. And then the overall, like we did a lot of analysis, like mapping the active site. So calculating the distances from each of these atoms, and then plotting and understanding that how these are being changed from the native and the mutant. And then like understanding that how these changes are there in the chains of these molecules, not only at a local level, but also at a global level. So from a local level, how do you see the changes in a global level? So these all are could be done through simply using some of the tools that we are going to cover in our upcoming program, structural biology program. But yes, I mean, it's something that anyone can do if with some guidance, obviously, and understanding like how things are, connected to, that is very important to understand that how local events lead to a global event, and how to map these changes, and give us solid scientific evidence to the community that this is how it works, and tell this everyone. So this is what I'm talking about in the next image. The next image is an example of an analysis. So here what we are trying to do is to map the sequences again to based on their movement. So this is a dynamic movement is being captured through PCA, and in this you will see positive and negative correlation. So there's a comparison of three structures here, and in this comparison what networks are derived, where residues that are colored in a clustered in the same group are colored alike, like the red ones. So by this you can try and understand that how the dynamic cross correlation between residues and the entire structure has changed between one structure to another. And that sort of comparison, when you're doing comparison at a local level, and also seeing how the changes are affected, and what are the residues who are responsible for these changes, and how they are doing it, because their interaction map is changed. As you can see, the image changed from A to B to C, and there are slight changes that you can observe when you look closely in the map also. So these slight changes the characteristics of the entire molecule, its function, and this is something that we wanted to map in the study using machine learning and also these techniques. So I wanted to show in this case study to you is that how different types of analysis can give you different kind of results and new discoveries in this area, and which is quite possible to do in many other aspects. So also, like, coming back to this another case study. So I think maybe we will be doing this in our next session when we meet again. So I'll just not go in more depth about the other case studies that I put together. But yeah, I also wanted to discuss with you some of these, like docking and all. So I think we can schedule another session soon. But yes, I mean, before we conclude today's session, I wanted to also tell you about this program, also some of these events which are upcoming, so that when we get the opportunity, then we learn about all of this. So from in March, we have a lot of events coming up, and many of you are welcome to join, because many of all the first sessions of these sessions will be free, and also many of them are completely free, like the OmicsLogic symposium. And I would request you also to share this with the others who could benefit from these upcoming events. Will be happy to share the links also of the symposium and also the workshops associated with the symposium. So there are guest lectures in these workshops that you'll find, and the symposium will have a lot of great speakers. In the next month, in May, we have programs coming in, like working with R and working with Python, space omics, infectious diseases. And this is the program that I wanted to tell you about, that is going to start in April 11th and May 11th, is the structural cheminformatics and structure biology. And before I conclude, I also wanted to tell you that I'm looking for students who are efficient and are good in structural biology to work on certain aspects and some projects. And this is not a paid internship, but this is like an internship that you can do with me and learn about many of these projects, how they have worked on, and how we can use this in different aspects. So thank you so much for inviting me for today's session. And I would like to pass it back to Rutvi to conclude the session. Thank you so much.
Thank you so much, Mohit sir, for this enlightening talk. I would like to take some questions. There are pretty much questions in the audience, so I'll just speak out to you. So the first question is, what are algorithms used to predict CA binding affinity?
So usually when the project started, there were no such algorithms. There were some algorithms which are not working efficiently. So this is one of the only from sequence. But yeah, from structure, if you know the structure of the protein, then you can predict binding affinity through molecular dynamic simulation calculation, free energy calculations, and some other methods which requires quantum calculations, quantum chemistry calculations. Yes.
Okay, so since we are running late, I would like everyone to ping in their questions to Mohit sir at his email address, that is mohit@pine.bio. For now, I would like to introduce you to the programs that Mohit sir already introduced that are coming in the spring semester. So I would greatly recommend you all to be a part of these programs because they are very fun learning, and especially for beginners in bioinformatics, this is the platform for you guys. So you can scan this QR code in order to register for the Omics Research Symposium. And the symposium is a free event to attend for all students or at all levels, including faculty, professionals, and experts in all background. The deadline to submit your poster abstracts is March 10, 2022. You can register yourself at the email, at the links provided in the chat box. And we at Pine Bio have different platforms on which we can connect with you. That is Facebook, Instagram, LinkedIn, YouTube, and Twitter. The links would be shortly shared in the chat box. And it would be really great of you guys to just scan and fill this feedback form in order to help us, improve in the future sessions. You can even tell us what kind of sessions you want in the future. And just to give you a